to continue our discussions in microlab. So we go now to another disinfecting agent or antiseptic agent known as iodine. So usually iodine is used in alcoholic solutions. So usually tawag doon called tincture of iodine or Lugol's iodine. So the preparation usually mayroong 5 to 8% iodine plus yung potassium iodide na pwede ring proportion is 10 to 15% and plus distilled water. Okay? Or kung sa dressing naman siya, usually iodine 10% plus an alcohol. Okay? Pre and post operative antiseptic. Usually this is Lugol's solutions or Lugol's iodine. Okay? This one is no longer recommended to disinfect minor wounds because usually it induces scar, tissue formation, and increases healing time. Okay? Usually, mayroon siyang dinidelaid because iodine try to react with the protein in the wounds. No? Pwede siya magkaroon ng natawag na remodeling of the tissues. Okay? I know if you try to remember what do you mean by remodeling of the tissues. So there is what we call a formations or other arrangements of tissues during the healing process. That is why there is a delay. Gentle washing with mild soap and water or rinsing this a scrape with sterile saline is a better practice for antiseptic process. Okay. Next, sodium chloride. So used in general cleanser, use antiseptic mouthwash. So this one is weak antiseptic effect with a osmolality or natawag na to na isotonic solution of 0.9%. Usually isotonic solution is prepared 0.9%. Normal saline solution yan. Okay? Sodium hypochlorite used in the past, diluted, neutralized, combined with potassium permanganate or known as Dakin's solution or modified Dakin solution. Yung term na noon, I remember that one during our chemistry. The teacher said as the common name is modified Dakin solution. It is prepared combination of the sodium hypochlorite plus the potassium permanganate or KaMnO4. It is now used as disinfectant. Calcium hypochlorite used in Semmelweis as chlorinated lime in his revolutionary effort, okay, against the childbed fever during the time. Okay, sodium carbonate has antiseptic and disinfectant properties also. Okay, next, we get the term as bactericide. So when you say antiseptic or antiseptic is just inhibiting the growth of the bacteria. Bactericide, this is an agent used to kill the bacteria already. So bactericide or bacteriocide, okay, and it is a substance that kills bacteria and ideally nothing else. So bactericides are either disinfectants or antiseptics or antibiotics, but usually yung bactericides are killing. So there are other antiseptics that they are just inhibiting the growth. For inhibiting the growth, it cannot kill really the bacteria. Okay. So asepsis, this is a technique which is used to keep all unwanted microbes from field of work. So observation or prevention of sepsis is called asepsis or aseptic technique. Usually is applied in microbiology. That is very important, especially in the preparations of culture medium to prevent cross contaminations. Okay, usually before we prepare the culture, magkaroon tayo ng face to face later on, hopefully by January. So pwede nating i-perform yung common preparations of the culture medium using the agar. Okay, agar plate. Okay. Antibiotic, this one is substance that produced by microorganisms prepared synthetically. That is synthetic, meaning a synthetic that is prepared from the laboratory. Kasi there are two types of diba? Two types of drugs. We have the synthetic drugs or three types, semi-synthetic drugs, and we have the natural drugs or biological drugs. Diba? Yung synthetic siya is prepared in the laboratory. We have antimicrobial action called antibiotic. So antimicrobial agent, so this is a chemical substance or agents which have antimicrobial action is called antimicrobial agent, but the antimicrobial agent are not antibiotics. Okay, usually mga antimicrobial it's not only bacteria, includes fungi plus bacteria and other microorganisms. That is antimicrobial. Okay, antimicrobial property. Sanitations refers to procedure which is used to reduce bacterial load from food and utensils. Usually, di ba, sanitize. So, hindi mo na recommended i-sanitize mo ng chlorine yung mga utensils nyo kaya pwede ka magkaroon ng poisoning dyan. So, usually, just water is very important in cleaning. Next. So, microbial contamination, it is defined as the attack on food or any other substance by undesirable microorganisms. That is contaminations. So, meaning, a food that is considered to be freshly prepared and nagkaroon ng contaminations, so usually mapanos na siya. Okay, so usually magkaroon ng food poisoning that is already, or food that are not well cooked, hindi siya masyado na naluto maayo. Okay, lalo mga spaghetti, di ba, padali lang. So usually pwede siya magkaroon mga staphylococcus pathogens. Okay, spoilage, undesirable changes in food caused by microorganisms is called spoilage yan. Okay? So, kung microbial contaminations, pwede pa rin na hindi siya na-spoil, hindi siya panos. Pero madali siya mapanos. Pero kung spoiled na talaga, pwede magkaroon ng mga growth of microorganisms like molds. Diba? Kita mo na, mag-molds na siya. So, that is already spoilage na, panos na, baho na. Pero ang microbial contaminations, hindi pa mangit sa panos, pero may contaminated na siya sa ng bacteria. Okay? So, later on, it will lead to spoilage na. Okay? Inoculum, the microbial sample which is taken from the food, materials, water, soil, plant, animal, or microbial culture in order to culture them in a medium is called inoculum. It's called an inoculum. Okay? So, ang inoculum niya, usually, samples na yan coming from the example, kung later on in the lab, so, require ako ng mga samples coming from the feces, urine, mga ganun. Okay? So, that is considered to be ang tatawag natin na mga inoculum. So, mga inoculation, it is a process technique by which an inoculum is transferred to the culture medium by using a inoculating loop or needle. So, yan naman ang tawag na inoculation. So, yung sample mo, okay, inoculum siya, transfer mo doon sa culture medium sa may agar plate later. Okay, I'll show you yung tawag na agar plate. Okay. Okay, this one is a culture media. So, makita natin, culture medium is substance having favorable conditions such as moisture, food, pH, oxygen content, growth factors to support the growth of bacteria. Kasi there are a lot of, later on we'll discuss about the environment wherein bacteria are going to survive. Example, kung sa taas at mataas yung manubo yung pH, considered to be below 6, or acidic environment, tawag ng acidophiles na naman. Okay, may mga mesophiles man. So, we will discuss that one later. So, based sa pH, kasi bacteria will survive in specific pH. Diba? Oxygen content. Okay? So, this is considered to be ang tawag ng culture media. Usually, makita nyo ganyan. Okay, this is already the colony of the bacteria. So, later on, hopefully, I can show you how to streak, zigzag, may mga pattern yan ng streaking of the culture medium. Okay? Next. So, culture media may be prepared from individual ingredients. So, may mga dehydrated powders available commercially. So, usually, in our lab, no, may mga culture media tayo dyan. Okay, the common one is the agar. Okay, the agar. So, powder yan. So, we have to formulate. So, madali man yung mag-solidify ang agar once na it exposed to a lower temperature. Then, doon na tayo mag-culture. Okay? So, store the required quantities which can be used 6 to 12 months. So, try to look also the expiration date. Store the media away from moisture kasi very sensitive yung agar. Once it is contaminated by water, so there's a tendency na magkaroon ng ano yan, bacterial contamination already or mag-react siya. Okay? Securing caps all containers tightly to avoid contamination. Kasi yung bacteria is already airborne. Nandyan lang yan. Viruses, di ba? Store in a dark, cool, and well-ventilated place because it is considered very reactive in light. Okay? Keep a record of receipt, opening on the media. So, yung mga documents, try to file. Discard dehydrated media that are either darkened or caking na siya. Okay, bilog-bilog na. So, that is already dehydrated. Kasi ang media may tubig yan. Okay. But this has small content of the media. So, later on, we will show you sa next topic natin about the percentage ng media. Water percentage. Rotate the stock of media following the principle first in, first out. So, dapat, kung sino yung first expiry, yun ang i-first move mo. Okay? O first mo na gamitin. So, for preparation sa media, adhere strictly to manufacturer's instruction. So you follow simple instruction there. Storage, usage niya. Prepared media should be protected from sunlight and heat because it is very reactive. Okay. Sino yung preparation yun ng ng bakal ka ng gulaman sa market? Okay? Try to hit that one. Di ba ma-liquefy siya? So this is also one example, yung mga commercially prepared, once ginhit mo siya, pwede siya magkaroon ng inatawag nato na may mga liquid portion dyan. Matunaw siya. Okay? Next, types of culture media according to the nature of constituents. Culture media may be divided into, we have natural media. So this one contains natural substances, chemical compositions which varies from time to time. Okay? And synthetic, of course, contains chemical composition, comprise solutions of mineral salts to which the substrates to be tested is added. So, yung mga substrate, usually, substrate usually, of course, the bacteria. Kasi yung bacteria or the microorganism there live in different salt content. So, hindi pares-parehas na yan ang mag-culture ka. Okay? Next. On the basis of nutrition, so we have ordinary media. So, this one, amount of nutrient substances for the growth and propagations of microorganisms. For example, nutrient agar and nutrient broth. Okay? Mag-nutrient agar, ya, solid yan. Mag-broth yan, parang solution yan. Okay? Enriched in isolation media, this may be example nutrient media containing sufficient amount of essential constituents, growth of bacteria such as blood agar and serum agar. Kasi blood agar and serum agar contains also protein, kasi one of the important nutrients that are being utilized by the bacteria to survive is the protein. So kung serum agar siya and blood agar, almost the same lang yan. Okay? Pero yung blood agar, syempre, ang serum, separated na yan na liquid coming from the blood. Okay? But both medium contain a protein. Next, selective or inhibitory media. This one. This media contains such substances which help the growth of only one type of bacteria and inhibit the growth of other bacteria. So, tawag yun inhibitory. Meaning, yung Staphylococcus aureus lang siya, other bacteria cannot grow there. Okay? It is only Staphylococcus, o Proteus vulgaris lang siya, Proteus lang. Okay? And we have also our indicator media. Generally, consists of a sample, but nutritionally adequate base to which substrate and indicator are added to show the change in reactions occurred. So, this helps identify bacteria or other microorganisms. So may yan, may mga color yung reactions ng mga substrate nyo, okay, or the bacteria that is being added, plus yung mga indicator. Kasi you have the indicator, yun ang example, color na agar mo is orange. Once you added the substrate, okay, you added the sample of that bacteria, it changes to black or it changes to brown, so that is already indicated. So based on that color, sample, alpha hemolytic or beta hemolytic, example, yellow siya, di ba? Kung yellow siya, beta hemolytic siya. Kung brown daw red, I think brown is alpha hemolytic. So this is considered to be a blood agar. Okay. Sorry, that is already considered to be an indicator media pala. Okay. Next, solid media. These media are widely used in laboratory. This is very common advantage of their use. So one can see the bacterial colony on the agar medium, and colony characteristic helps determination and identification of many bacteria. So this is considered to be a solid media. So kung solid siya, this is very common. Ang pinaka-common na encounter tayo usually is the agar. Okay? So makita niyo yan ang mga colony, form yan. That is already a colony. Okay, meaning the bacteria already grow because of the nutrients that is present from that medium. So we have liquid media. So this one called broth. So bacterial colony cannot be observed in this media, but this media is used for study of biochemical characteristic besides samples of pathogens that can directly place in this media. Usually, it's difficult to identify all types of organisms suitable for isolation of bacteria and blood culturing and water analysis. Usually, sa liquid medium man niya, usually identification of the change of the color of the medium. Okay, sa liquid. So, usually kung cloudy na siya or more cloudy siya, for example, makita mo siya, nagbutang ka ng sample mo dyan sa liquid broth. So, kung more ang turbidity, tamaan ka turbid, tamaan ka cloudy na siya, meaning there is a presence of bacteria. So, that is already identification. Okay? Next. So, media constituents. So, we have agar. This can be obtained in shreds, flakes, granules, powder is made from certain types of seaweed. So, yung seaweed, carrageenan or agar-agar that is considered to be a polysaccharide. Later on in biochemistry, that belongs to a polysaccharide. It is a gelling properties used to the preparations of solid and semi-solid culture media. So, this is very common. It is insoluble in cold water but soluble when heated about 900 degrees Celsius. It is become solidified at below 450 degrees Celsius. So usually makita mong agar. Try to buy agar gulaman. Try to heat that one. Ma-liquefy na siya. Hasta na siya kung tamaan mo kay heat, hasta na siya liquefy. Pero once the temperature subside, automatically it will solidify. Okay, that is one characteristic mo agar. Hindi mo bakal ng agar powder. So kung gusto mo lang siya i-test, so pwede na yung agar na carrageenan para mabalaan mo kung pila ka degrees siya liquefy and solidify. Okay? Next, we have peptone. So it is a product of varying composition of enzymatic hydrolysis of animal, vegetable, protein from such as muscles. Okay, materials such as muscles. Usually muscles contains a lot of proteins. Liver also. Blood, milk, more on fats. Casein, this is considered to be a protein. Lactobulmin, protein. Gelatin, and soybean. So mostly, ang peptone are composed of a protein. So exact composition depends on the raw material and the method of manufacture. So depende yan sa manufacturer. Okay. There is what we call a common supplier known as Charlau. So they are the one that are considered to be high quality manufacturers of the tawag nito mga reagents, including of course the medium. Okay. Next, we have meat extract. This one, commercial meat extracts usually by protein degradations products, protein degradation products, vitamins and minerals. So beef heart, liver, brain, spleen, etc. used to prepare meat extract. These extracts are really available for easy to use. Powder niya siya actually So usually yan ang combination. These are nutrients of bacteria these are nutrients for bacterial culture or bacterial growth. Yan ang kailangan ng bacteria to live. Okay? Next, we have yeast. So, yeast extract is made from bakers or brewers' yeast, a rich source of amino acid, again another protein or amino acids, vitamin B complex. Culture media, it is used for supplement and replace meat extract. So, kay mahal man yung meat extract, usually mahal yan ng powder. So, pwede na yung yeast extract niya. Okay?